Okay, Dean, you're welcome to start.
All right, well, thank you for attending this session. Today I'm going to talk to you about my work on behavioral plasticity and courtship and fruit flies. And before getting started, I just want to acknowledge the majority of these middle authors are students who took a research class with me and who did a lot of this phenotyping. All right, sorry, some technical difficulties. So my lab works on speciation in Drosophila. So this is just a small snippet of the diversity you see in Drosophila. And while a lot of it might be due to ecological differences, we know a lot of the diversity we see is due to sexual interactions and mating diversity. And when we think about mating interactions, I want to bring up two kind of major patterns that we know about in fruit flies, but other animals as well. And that is when we kind of see a species making a mate choice. So here I have it outlined in this black box. We kind of see a correlation of traits that, there's a female preference, it matches some specific male courtship traits, and these will differ over populations. We use courtship and these sexual characteristics and kind of secondary sexual traits oftentimes to define a species. So we kind of have this one main observation that courtship is species specific, and sometimes we consider that either stereotypical or fixed. On the other hand, we know that mating behaviors and a lot of other social behaviors are also plastic. So this is a famous example in fruit flies where females are kind of using mate choice of other females to determine their strategy. So I really want to start asking the question, well, how does plasticity affect the evolution of reproductive isolation? So that's a really big question. So for today, I'm going to start with kind of the first entry into this, thinking about what cues males are using when they're actually adjusting their courtship strategy. So the system that I'm using is Drosophila melanogaster. We do use a lot of lab type strains and genotypes, but we also use natural populations. So in this scenario that I'm going to be talking about today, we're using natural flies that come from Africa and outside of Africa. And the important pattern here is that if you take kind of a female from outside of Africa, it's in this pink color, and you expose them to a male within Africa, they mate really well. But the opposite is not true. These African females are very choosy. So they're making kind of decisions based on the different cues these males are giving. So what's different about these populations? A lot is known about kind of courtship differences. So a lot of labs spend time kind of watching what these flies do, analyzing their pheromones, including my lab. We spent a lot of time doing this. And when I started in this system as a postdoc, that was one of the first things we talked about. So this chunk of the experiment has already been published, but I want to bring it up because it starts the foundation of what we're working on now. So when I was a postdoc, we started with the question of there are a lot of differences. Which ones actually predict mating success? So we took kind of African and non-African females, exposed them to a panel of a diverse panel of male genotypes and just asked, how are they behaving? What does the mating patterns look like? Um, we were using kind of a very standard kind of approach to analyze behavior in melanogaster, where people again assume that this courtship is stereotypical. It kind of defines the species, with the most important trait being singing. So the male gains some olfactory or I guess gustatory information through pheromones, and then they perform this singing behavior. So that's what we focused in on. And those are data that I'm going to show you right now. So on this graph, I'm going to have kind of the results of looking across this panel in terms of singing. And on this column here on the left-hand side, it's a non-African female. On the right-hand side, it'll be an African female. So this first point I'm showing you is a non-African male, a non-African female. These would be standard lab strains. They sing a lot. That's kind of known in the literature for a long time. But what was surprising to us in the study is that singing was highly variable. Some of these flies hardly sing at all, but they're still successful in mating. So that was kind of an interesting result. But what I want to focus on today is that singing was plastic. So we define plasticity as a change in phenotype due to the environment. And here, the environment is the female that's being courted. This is the same male genotype has this plastic response, and it's pretty uniform across all the genotypes where they kind of down regulate singing when they're exposed to an African female. So the question that I'm going to ask in the talk today are what cues are males using when they're kind of adjusting their singing in this way? A great candidate is pheromones, cuticular hydrocarbons. A lot has been done in melanogaster to identify the specific cells on the foreleg, the foretarsus that are picking up chemicals. We know genes involved, we know chemicals involved. This is a 29 carbon compound. And a lot of really good work in other labs has actually connected kind of this stimulus to a neural circuit in the brain that projects to kind of a descending neuron that can actually controls the motor neurons involved in song. So this is where we wanted to start. You know, singing is different. It must be due to differences in female hydrocarbons. And luckily for us, African females indeed have very different hydrocarbons. So in this table, I'm kind of showing you the different populations of flies and their hydrocarbons. This red one again, or pink, is the standard lab fly. Their main carbon compound is a heptacosadiene, so 29 carbon with two specific bonds. The African flies, they produce 711, but they actually produce their main compound is an isomer, so just the position of the double bonds changed. Interestingly, and this is work again that's been done a long time ago, it seems like there's a naturally occurring a null mutation in non-African flies. There's a deletion in a gene that kind of makes the shift in this hydrocarbon. So this is where we want to start. We want to test these CHCs, but the problem is that behavior is kind of confounding these hydrocarbon differences. As I tried to introduce the system earlier, non-African flies, they are not choosy. African flies are very choosy. So the first thing we did is try to decorrelate or decouple these two traits. So we actually use CRISPR to make a DSAT2 null fly in an African background. So this is kind of the gene structure of DSAT2. It's a pretty simple gene with only one isoform. We just cut out the whole gene, including some of the upstream regulatory regions, replaced it with a visible marker. So basically now we have an African fly that has a DSAT2 null mutation. I'm not going to show behavior data on this fly today, but it is still choosy. We've kind of verified that, and we switched the hydrocarbons. So those are the data I'm going to show you first. So what I'm showing you now is a zoom in on a chromatograph. So this is kind of coming off a gas chromatograph, which separates molecules by size. I've just zoomed into this region that we care about. This black dotted line is our standard African fly. So its main compound is 5,9-HD. It does make 711 at a smaller amount. This is an internal standard that we use just to measure quantity. The blue line is our null fly. So you can see we've been pretty successful. We've knocked down this compound completely. We've shifted this up. So this fly now looks like a non-African fly. If we actually quantify that, you know, there's a significant decrease in the 5,9, a significant increase in 711. This red fly is the standard kind of non-African fly. So there's no difference now between these mutants in terms of their hydrocarbon. So now we use this fly in a behavioral assay. So we give an African male these three different types of females, and we just ask, how are they going to behave, right? We now have this tool we can use to really test whether pheromones are controlling this plasticity. So we have a standard African female, a non-African female, and our mutant. The mutant was created from this African female. So they're genetically identical except for at that DSAT2 locus. And we video recorded all of these interactions so that we could measure some of behaviors that are common in African flies, like scissoring and chasing, as well as kind of the main behavior that people pay attention to, the singing. We analyzed them two ways. One was using kind of machine learning. There's this framework published in a paper, they call it Dance, where it's using fly tracker and Java, so pretty standard tools. But some of these behaviors, we don't really have great classifiers or training data for them, especially things like scissoring and grooming. So this is where the undergraduates in the class came along because they did a lot of kind of manual ethograms of these behaviors to quantify them. So what I want to show you first are kind of what the data look like for these African flies. So on the bottom, I have kind of just the genotypes, like what their background genome is, and then what their genotype is at DSAT2. And then I'm showing you the singing behaviors first. So this is singing. So here's the cartoon of what the fly would be doing in this part. African flies, they don't really sing a lot to other African flies. This behavior on the right-hand side is called circling. It's really phenotypically similar. They are making a song, but they just move in an arc. So this is kind of our baseline. This is how an African fly treats another African fly. When we look at the non-African flies, we do see an increase. It is statistically significant. So, we're seeing this plasticity, and now we can ask, what's happening with this DSET2 null fly? It turns out that they're being treated like an African fly, right? They have the pheromones of this red fly, but that's not how they're being treated. They're being treated just like another African fly. This was really surprising to us because we thought this pheromone change was going to really kind of dominate our signal, but it suggests that they're paying attention to other feedback traits. The one other piece of data that I do want to point out that I think is really interesting is that not only did we look at the time spent singing, but the number of songs that they're performing. Um, and this blue mutant, they actually start singing to them, then stop. So maybe it is they are getting that initial pheromone cue, but then subsequent female feedback is kind of telling the male fly, okay, this is an African fly, not a non-African fly. So there's a lot of variation in this phenotype, but there is a significant difference in the statistical models. So again, it looks like males are relying on other cues. We know females perform rejection behaviors and receptivity behaviors, so we're going to start kind of looking at those female-based behaviors. And why is this important? So these are two papers that have really been kind of influential for me thinking about the evolution of sexual isolation. And the main point of both of these papers is that there's a lot of asymmetry. When we look in kind of real systems, we notice asymmetry. That's happening in my system with these African flies. So I'm starting to think that a good working hypothesis might be that it's not the loss of a signal that is important for asymmetry, but maybe it's this plasticity. We see kind of a shift in a mean trait, a shift in some type of plastic response that could be leading to asymmetric reproductive isolation. And that would make sense if courtship plasticity is adaptive. And so I just want to return to this result I showed you previously, kind of first showing you plasticity. There is a mean kind of shift. These are non-African flies. They sing more. They are still plastic, but they're not really reducing their singing in a way that's acceptable to African females. We know that they're rejected. So that's kind of my working hypothesis that maybe over time there was a mean increase, plasticity is maintained, and then to describe longer term speciation, that plasticity might erode over time. So with that, I'd like to thank a lot of the people in my lab who helped with this work, other labs for supporting this work, and I'm happy to take any questions.
Thank you, Dean. We have about 2 minutes for a Q&A session. Does anyone have any questions? If not, I'm gonna ask one question. Is what, sorry if you have already repeated this, but like what would be the next step in this study?
Yeah, that's a great question. So for us, we're going to kind of approach this a couple ways. One is going to be trying to determine the sensitivity of males to different compounds. Like I was suggesting at the end that maybe males have upregulated singing and might lose plasticity. Well, we can actually get these pheromones synthetically, and we can kind of put them onto a fly that doesn't have any pheromones. So I think that'll be for me a good step. Like, what's the sensitivity of these males to elicit singing? The second thing that we want to do, using that mutant and kind of using the males we already have, is to think about that adaptive part. Like, I suggested maybe it's adaptive, but if we put these males in competition that have plasticity, what's their kind of rate of acquiring mates? The videos we looked at are just two flies, one male, one female, but the dynamic might change when we have kind of multiple mates and a male that kind of is not plastic and just mates versus a male that's kind of trying to get information before making a mating choice, as well as the female making a mating choice as well.
Thank you. All right, our next speaker, please start sharing your slides. and you're welcome to start.
You, I will present my project about foraging activity, cuticular hydrocarbons, and water stress management in the red harvester ant. Beginning with a word about the model species, Pogodemyrmex barbatus. Sorry. It lives in arid habitats of Mexico and the United States South. And here is a picture of the field site in which the Gordon Lab runs a long-term study running for almost 40 years. And during this study, the team censored every nest in this field site, and that amounts to a bit over 300 colonies per year. So several colonies have been censored through their whole lifetime. And it's worth noticing that over the last years, there is a drought, an intensifying drought in the area that results in reduced food supply for the ant. Of course, from such a long study, a lot is known about the population. What is relevant for my project is that in dry conditions, some colonies reduce their foraging activity. Instead of going out and looking for food, the ants stay in the nest, so they sacrifice food intake to conserve water. But not all colonies do that. Some colonies maintain foraging activity even on dry days. It is also known that colonies that reduce foraging and the ones that do not, they differ in their CHC profiles. This is interesting because one major function of CHC layers in terrestrial arthropods is to limit water loss through evaporation and to protect against desiccation. Specifically, the colonies that reduce foraging in dry conditions, they have higher abundance of alkanes and higher abundance of short chain analcanes. And these compounds are not the best to protect against desiccation. They permit water loss. So based on this previous knowledge, the rationale for this project is that CHC differences will explain water loss differences, in turn explaining survival differences. And we formulated the hypothesis that colonies that reduce foraging or not would differ in their response to temperature and humidity conditions, so that there is an interaction between the foraging activity and environmental conditions. Not all colonies will respond similarly. And we conducted a CHC analysis, a water loss assay, and a survival assay. To do so, we started by collecting ants from 24 colonies, from which we know the foraging activity. And we collected at least 36 ants per colony that we split in 3 different acclimation treatments. Cold and humid, that is the mildest conditions. Hot and dry are the harshest conditions, and hot and humid are intermediate. After an acclimation time, the ants were used for CHC analysis or a water loss assay or a survival assay. For the particular hydrocarbons analysis, we analyzed them using a gas chromatography mass spectrometry, and we analyzed the whole profiles, not specific compounds or specific classes of compounds, but the whole of it, using a multivariate test and focusing on some factors of interest, the acclimation treatment, and the interaction between the acclimation treatment and the foraging activity. Here is a visualization of the results. You see that the different profiles are quite similar, and there is a lot of overlap between the 3 acclimation treatments, confirmed by the statistical test. Treatments do not differ, and there is no significant interaction between the acclimation treatment and the foraging index, the foraging activity. Sorry. So for the water loss assay, we isolated ants, exposed them for 4 hours to the harshest condition, the hot and dry condition. And the difference in mass between the beginning and the end of this assay, we assume that was all water loss through evaporation that we analyzed using a linear mixed model, the same factors of interest. Here's the visualization of the results. This graph is slightly counterintuitive because on the left are the colonies that maintain a high foraging activity in dry conditions, and the colonies reducing their foraging activity are on the right part. You see that ants from colonies that reduce their foraging activity, they tend to lose slightly less water, but this effect is not significant. What is the difference between different treatments. Specifically, ants acclimated to the cold and humid conditions, they lose more water than the ones acclimated to hot and dry conditions. So the ants previously exposed to hot and dry conditions were better prepared to this assay, and they lost less water. But you see that the slopes of the 3 regressions are almost identical, so the interaction between the acclimation treatment and the foraging activity is not significant. And that leads to the last part, the survival assay, during which we isolated ants, and they were exposed for up to 13 hours to the hot and dry conditions. We monitored the ants every hour, and if one ant did not move at all, it was recorded as dead. And the number of ants alive was used for the statistical test. And once again, we used the same factors of interest, acclimation treatment, and the interaction between foraging activity and acclimation treatment. Here is the visualization of the results. You see that there's more going on. The 3 acclimation treatments are not different, but quite clearly the slopes of different regressions are not the same, so it's no surprise that the interaction is significant. So different colonies with different foraging activity, they do not respond similarly to the acclimation treatments. Specifically, ants from colonies that reduce their foraging activity, ones acclimated to the cold and humid conditions, they have the highest survival rate. The ones acclimated to hot and humid conditions have a lower survival rate. For ants from colonies that maintain a high foraging activity on dry days, the trend is reversed. The ants acclimated to hot and humid conditions had a high survival rate, and the ones acclimated to cold and humid conditions had the lowest survival rate. So to summarize, the CHC profiles are very stable. The ants acclimated to harsh conditions lose less water. And the colonies that reduce foraging activity or not differ in their survival rates depending on the acclimation treatment. So our initial rationale that CHC differences will predict water loss differences, predicting survival differences. The data show that it's not as simple. Other factors are likely to play a role in this system, and we will continue to investigate this. It's still an open question. It's important to predict the future of these populations as, as their ability to adapt to this intensifying drought. It is known that until 2010, the colonies that reduced their foraging activity had a higher reproductive success. But with changing conditions and importantly reduced food supply, will these colonies be able to maintain their advantage is an open question. And I would like to thank collaborators from the University of Mainz, my supervisor who also secured a grant that funded this project, direct colleagues from the department, everyone who joined for fieldwork and helped collecting samples and data for this project, and of course, thank you for your attention.
Thank you. Does anyone have a question? Yes, Maya, please go ahead.
Hi, awesome talk. I'm so curious, just because I'm interested in this interaction that we see of like getting nutrients versus saving energy and things like that. Has there been a lot of work done? And you may have mentioned this, I apologize doing tech support here, but have you looked at whether there's been work looking at like the rest or what rest is taking place, or whether there is rest or like a sleep state in these ants, and how that might be like a trade-off of resting versus being outside in addition to like reducing that moisture loss? And thank you again.
Yes, that's the overall framework for what my PI is doing for several years. The, the background idea is that ants they lose water by foraging, especially when it's very dry, but they do not drink liquid water. They gain all their moisture from their food, from the seeds they eat. So depending on the conditions, they regulate their foraging activity, and they may reduce food intake but save water. It's always considered at the colony level, and to my knowledge, it was not investigated the individual behaviors. If some foragers rest more, I cannot tell you that.
Thank you.
Thank you.
Oh, Aurelia, we're out of time. Can you put your question in the Q&A? And then our speaker can answer your question there. So our next speaker, Quinn, please start sharing your slide.
Hello, everyone can hear me?
Yes,
See
we can hear you. And let's wait for like about half a minute. So
All
we'll
right.
start on time. Yep, you're good to go.
All right. Hello, everyone. I'm here to talk about my work showing that outbred Pukeko achieve higher dominance scores within their groups. So first, I'd like to introduce you to my lovely bird. This is a pukeko. It's found in New Zealand. Um, we study a population in the North Island that we've done a fair bit of work on, and it's been shown that they're very unique, or there are a couple aspects of their biology that make it very interesting for us to study them. One, they form these very stable groups typically, and within these groups, they form these very transitive dominance hierarchies. These groups are also very phylopatric. So once a bird is laid, an egg is laid into their group, they usually stay with their group their entire life after hatching. Within these groups as well, they are known to be joint laying. So joint laying, if you're unfamiliar with the term, is simply when multiple females lay eggs in a shared nest, and it's relatively uncommon among bird species. Within these groups, we also know that there's a very high reproductive skew. Typically, within these groups, the most dominant females, like 1 to 3, are the ones that are laying the eggs, whereas most males are able to, in theory, participate in the copulations and the production and siring of future offspring. Now, the keen listeners among you may have noticed that I said they stay with their groups their entire life after hatching. And so when they started studying this species or the subspecies back in 1970s, they said, hey, they're there their whole lives. What's happening? This must be pretty rife with inbreeding. And that is what they found. So when they color marked birds, they found that 75% of copulations were between first order relatives, so mother's sons, father's daughters, full siblings. And that was shown to be pretty unique among this population or this subpopulation that we're studying. So jump ahead 40, 50 years, comes to me and our lab. We wanted to look at the consequences of this inbreeding because they're still around. We still see them and these populations in our territories that we're studying. And what we've seen is that when we compare populations that are outbred, that are different from our study population, we see that the hatching success in those outbred populations are higher compared to our inbred study population. And when we look at our inbred study populations, when we look at those clutches that have some that fail, some that hatch, we see that the ones that fail are typically more inbred. We see them having higher inbreeding coefficients. So knowing that inbreeding should, in theory, just dogmatically, we think that inbreeding should have some impact on success, and knowing that we have seen some impacts on their success from inbreeding, we wanted to look at the other traits that may suffer from inbreeding depression. And so because dominance has impacts on these breeding dynamics within these groups, we hypothesized that dominant status within these groups would show evidence of inbreeding depression. And so with this, we predicted that we would see that outbred birds are achieving higher dominance scores than the inbred birds within their groups. So what that looked like is we went down to our field site in New Zealand. We got every bird. We found a bunch of groups, about 20 groups, that we were able to capture all or most of the birds in these groups. Group size varied from between 5 individuals up to 14 in this study. And then we banded as many as possible so that they were visually identifiable. While we were banding them, we also took a blood sample. Once we'd had every one of these groups banded and visually identifiable, we then started observing them. We would observe them for 30 minutes over 30 minutes a day for 10 days straight. And we would record them with a Sony Handycam. We'd then review all those Sony Handycam videos to classify every interaction that we could within these groups. So pioneering work done by John Craig, also in the 1970s, established an ethogram that we were able to use that classified all behaviors into this dominance framework, if possible. And so if we saw a bird do one thing, we knew that this bird was dominant. And if we saw another bird do another, we knew that that was a subordinate bird in that interaction. And so we classified all of those interactions that we saw over these 5 hours over 10 days. And we were then able to calculate a dominance score for them. So just a snapshot of what that sort of looks like. I hope the video is working. We have these birds foraging in this area. And then we see one bird start pecking the other, more aggressive than the other. And then they sort of like square up and start sizing each other up, showing each other their shields. And then we took the identification of each of those birds. And then we were able to record that one bird was dominant, one bird was subordinate. You can kind of see on this still right here that these birds have these these color bands on their legs. So once we'd done that, we were able to calculate the David scores, and then we came home to our university in Canada and we took those blood samples with us. We extracted the DNA from the blood and amplified 13 different microsatellite loci and genotyped them. The data from those 13 different loci were then input into the software EM-IBD9 to calculate our inbreeding coefficients. This software also calculates standard deviation around this calculated inbreeding coefficient. And so with that inbreeding coefficient and standard deviation, we were able to simulate 1,000 values to sort of estimate the uncertainty in our models. We took all of these values, these 1,001 values that we've gotten, and we input them all into linear mixed effects models. And then we were able to see the impact of sex, of inbreeding coefficient, and the interaction between sex and inbreeding coefficient on their dominance within these groups. So what we found was that David scores decrease with increasing inbreeding coefficients. So a higher David score means more dominant within these groups. A higher inbreeding coefficient. Inbreeding coefficients go from 0 to 1, 1 being completely clonal, 0 being complete from a product of a mating that was completely unrelated. And we see that overall, David scores decrease with increasing inbreeding coefficients. So they get worse at being more dominant the more inbred they are. But we also looked at the interaction between sex and inbreeding coefficient. And we saw that in females, this trend holds true. You can see it in the figure on the left there. But in males, we see that there seems to be an almost reversal of the trend. There's a lot of uncertainty in our estimates. You see those blue bars in the background are the simulated values. We see that while the trend seems to go upward, it's kind of hard to say for sure which direction it's going. So we're not confident that inbreeding impacts dominance in males, but we can clearly see it that this is the case in females. We also looked at their within group rank. David scores are a bit more esoteric. For a bird, it might not matter if you're have a 10 times greater dominance or David score if you're still number 1 and number 2 in the group. So we collapse those all to just their ordinal rank. As a reminder, you want to be number 1 in ordinal rank. You want to be number 2. You want to be number 3. You don't necessarily want to be number 10. So we see that dominance action or more dominant birds are have a lower number for their within group rank. And we see again that trend holds for females. We see that their dominant status is higher, or the dominant status is lower as their inbreeding coefficient increases in females. And in males, while we see that the trend is the same way, we see that it seems to follow that same trend in males this time. Again, there's so much uncertainty in the values and in our estimates that we can't say for sure with confidence that we see this the impacts of inbreeding depression in males. Again. So all in all, we saw that female dominance seems to be more impacted by inbreeding depression than in males. And so looking back at the literature, this seems to make sense. In this species, part of their life history, females are more able to monopolize reproduction. And so it makes more sense that inbreeding depression would impact this trait in females compared to males. In domestic chickens, it's also been seen that males that are more inbred have lower dominance compared to outbred males. And conversely, in chickens, we see that one male is able to monopolize multiple females. And so that makes sense. We see the same sort of relationship there, even if the sexes are reversed, because the sex that is able to monopolize reproduction is the one that is more impacted by inbreeding depression. Overall, this seems to have some strong implications for this species. As I mentioned, there's been it's been studied for a long time that there's rampant inbreeding. And so if these groups or the subpopulation is able to sort of mitigate the impacts of inbreeding by the outbred birds being more successful, then it could be that the outbred breeders end up being more genetically diverse. Their offspring are less likely to receive these or to inherit these deleterious recessive alleles. And so their offspring will have greater success and sort of maintain a higher degree of genetic diversity within the subpopulation compared to if they were all fully inbred, just inbreeding rampantly and no impacts on their success. So with that, I'd like to thank specifically Ngati Manuhiri. They're the traditional owners of the land that we do our work. They've been very gracious to us and helpful in setting up and maintaining this study for us, as well as many people listed on this slide who have helped in some way or another that I'd like to thank. With that, I'd like to ask if there are any questions, and thank you all for your time.
Thank you. That's a very interesting talk. I actually have a question. So when you mentioned like you essentially use these video footage to determine like their dominance like outcome, I just wondering like exactly how do you tell who is dominant over who?
Yeah, so like I mentioned, John Craig did like he watched Pukeko for like 7,000 hours in the 1970s, and he basically classified the outcomes of every possible interaction that he observed. And so he knew that like if this bird did this and this bird did this, like that was sort of like a mating thing, or this bird did this, then it was just affiliative. And so we knew that if a bird did so like so and so posture, and a bird did this in response, then they were interacting, and one was interacting in response to the other. And the one that, yeah, and Dr. John Craig also noted which one was more dominant and which one was the more submissive behavior or posture. And so we were able to use sort of his ethogram that he set out to classify who was dominant in that scenario. And then once we'd done that, we were able to tally up the numbers of interactions, and those were fed into the David score calculation. The David score calculation from DeVries 2006, DeVries et al. 2006, I should say. Basically takes into account the relative proportion of wins that you have against an individual, and the relative score that those individuals have that you're winning against, with the idea being that if you lose to someone who's always winning, then it's like no harm lost, like no harm done. But if you lose to someone who's like really low on the totem pole, then that's like, oh, that's really bad. Like this baby beat you up, or like the bottom of the bottom kind of beat you up. So we use all those values to calculate that David score. Yeah, does that answer your question?
Yeah, that's very helpful.
Thank you.
Do we have any more questions? Dean has a question.
Yeah, this might be a little bit out there, but I'm wondering in a population that's more inbred, they're more close kin. So I'm wondering if like just less um, conflict interactions might be happening in those populations, which might be contributing to you like not seeing like an effect of inbreeding on dominance. Does that make sense?
Potentially, yeah. So I have done some like just like really coarse analyses looking at like if this group, like the average relatedness in X group, is lower, or yeah, if the average relatedness is higher in a group or lower, like do they have more aggressive interactions? Do they have fewer? And we do tend to see, like this is all preliminary, like I haven't finalized it, but we do tend to see that more closely related groups, or groups that are on average more closely related with each other, tend to have um, less of a difference in like the highest ranked David score and the lowest ranked David score. So like they do seem to be fighting less, or they're more egalitarian overall within these groups. So that could be something that's impacting it. We do like, because we are seeing it clearly in females, but we're not seeing it clearly in males. It could be that that dominance in males functions differently than in females. Like I said, like in females, it's the dominant one, two, or three that are breeding, but in males, any of them can in theory breed. It's also been said that males are typically the ones that are doing territorial acquisition and territorial defense. So it may matter more for males to be better at acquiring territory than, say, like getting a higher dominance rank. So that's something that we could look at, and we're kind of like banging our heads together, me and my PI, to see if we can get blood out of a stone with that one. But yeah, there's a lot going into it. Kin's like the fact that they're kin based groups is something that we're considering and trying to tease apart. Good question. Thank you.
Thank you.
Thank you, Quinn. I'm gonna have the last speaker, Owen, put up their slide.
And Owen, just double checking, you don't want to be recorded, is that correct?
You can record me. I clicked that by accident. I don't care
Okay.
if that's easier for you.
Great.
Yeah.
Yep.
Thanks for checking. All good? You can see
You're
me?
welcome to start. Yep.
Okay, cool. Thank you. Hi, my name is Owen. I am a master's student out of the Lung Lab at Laurier in Ontario. And I'm going to talk to you about my research that looks at the relationship in between sexual dimorphism and diet choice and resource partitioning in fruit flies. So I think when we think of sexual dimorphism, we think of these really obvious classic examples, like elephant seals, where males are much, much larger than females and they fight each other for access to mates, or peacocks, with the males having these elaborate trains and they use them to attract females. And these all have a benefit to reproduction. But there's also other dimorphism that evolves that's not necessarily related to sexual selection or reproduction, more so diet choice or and the way they use their environment. So an example of that would be oyster catchers, which they're a shorebird. You can find them in Australia. On average, males or females have a 90% longer bill than the males do, and this translates to a diet difference as well, where females tend to prefer more soft-bodied prey and males like more hard-shelled prey. And males use these rocks called anvil sites where they smash open the hard-shelled prey to eat, and females rarely ever use this. And another example is northern gannets. These are monomorphic seabird species, and they forage by diving in the ocean for fish. And studies have shown that females actually dive at deeper depths and different times than males do. So both these are examples of different sexes utilizing their environment differently, either through morphology or through behavioral difference. And these are kind of what are known as ecological sexual dimorphism. So it's dimorphism that has to do with how they use their environment. And this has been talked about for a long time. Darwin mentioned this in Descent of Man, but very briefly. And it's kind of been built upon by other scientists since then. Like right here, this is a model from Slatkin from the 80s. This is based off of his ecological character displacement model, and he kind of tweaked it to be more so as in interspecific competition as opposed to interspecific. And he's just showing here that there's if there's two different phenotypes, a male and a female phenotype, and they can specialize and use their environment differently, they can kind of increase the carrying capacity of their total environment. So not only does it benefit the individual sexes, but also the population as a whole. So looking at these models and these examples we have, there's many more examples. We know kind of theoretically how this happens, and we have examples of it, but we are really missing empirical data to link the two. We don't know what factors kind of contribute to this, ones contribute more, what the rate of this is. So the really only study that has been looking at this empirically was done by Stephen Delisle back in 2022, where he took fruit flies, which we know are a generalist species, but they have these sex-specific diets where they can kind of optimize their fitness, where females tend to have want more protein for oogenesis, and males want more carbs to fuel their really expensive mating behaviors. So knowing this, he took fruit flies, he took 8 groups specifically and split them in half. So 4 went into these high competition, low nutrition environments, and 4 went to this high nutrition, low competition environments. And he fed them through these little microcapillary tubules here with this kind of liquid-based diet. And they had a choice between a carb-rich liquid source and a protein-rich liquid source. So he figured that they would be more choosy. So this is his ancestral population here. So you can see this is the diet content. Males are blue, females are pink, and they're pretty overlapping. And then after only 3 generations, he found that in his high competition environments that there was this divergence in diet, where females tend to eat more yeast to protein or yeast to sucrose ratio, which is really interesting. So this is kind of the only study that looks at this empirical link, and it was really, really impressive. So it kind of inspired me to build upon this. So what I did was kind of go over Delisle's work and use the things I thought I did great, and maybe look at some things that were kind of made things more unclear and what I wanted to build upon in my study. So Delisle used one population of fruit flies, the LHMs, and there's only a 3 generation study. Whereas for mine, I used LHM and IV fruit flies, which are a cousin stock. So they have different culture cycles and densities, and they're from different origins. I also ran it for 13 generations as well to just get more of a overall trend. He compared things to his ancestral population as opposed to a control group, whereas I used control groups for each population to see if the environment was actually doing anything as opposed to the diet. And I also used competitive control groups to test fitness against. And then due to the way that the micropillars work, he used, this liquid food, which is unfamiliar to the flies, and the whole delivery system is different to what they usually have. So I gave them kind of similar food to what they used to, just kind of in different ratios. And again, the way that these capillaries work, only one fly can feed at a time. So sometimes this leads to larger bodied flies or more aggressive flies bullying off others feeding. So that can lead to traits that we don't really want to select for. So again, I used a similar delivery system that they're used to with access for a lot of flies at a time. So this wasn't an issue. So this is kind of how I set things up. This is one of my control cycles here. So I had 3 controls for each group, 3 for IV, 3 for experiment, or 3 for LHM. And you can see here they start each cycle with fruit fly eggs in these vials. The eggs would hatch and the larvae crawl onto the sides of the vial here where we put in an acetate sleeve. And then they pupa it on the sleeve and we can take it out and put it into these population chambers. In these chambers, there's just what we call a food array with a bunch of food. And they sit in here for 6 days. And at which point we introduce new vials with standard media. And then they lay their eggs and then these start the cycle again. And this is my experimental design. It's identical to control. The only difference here is the food array for the population chambers. It is a heterogeneous food as opposed to homogeneous. So in the control, it is every patch is a good mix of carbs and protein. Where in experimental, there's a carb-rich food and a protein-rich food. It's just spatially separated. And importantly, it's the same nutritional overall number in each. So there's no less nutrition in each one. It's just kind of spatially separated. And I also had 3 experimental for each. So I had 12 population chambers in total. So to determine how these flies were reacting to this environment and this food choice, I ran some fitness assays at different generations, and I also ran behavioral assays to test how their diet choice was evolving. So for the female fitness assays, I took focal females individually into these test tubes, let them lay eggs and remove them, and we can count the eggs in our offspring. For the male fitness assays, we took focal red-eyed males and then placed them into these competitive vials with 5 brown-eyed male competitors and then 6 brown-eyed females. Let them mate, separate the females, and we can count the number of red-eyed offspring to brown-eyed to get an idea of focal male fitness. And then we did these behavioral studies where we put flies individually in these petri dishes and gave them a choice between this protein-rich and this carb-rich food source, and basically noted what they were on every 15 minutes for about 6 hours at a time. Okay, so this is a lot of histograms I'm going to throw at you here, but this is kind of what we saw. So this is male fitness. So IVs are on the left, LHM flies are on the right. This is looking at proportion of offspring sired by the focal male. And for all of our data, this is all bootstrapped, and we ran 10,000 permutations just to get a better idea of what was going on. So you can see here, we have this kind of not huge difference in IV males, and then unexpectedly, the control males did better in the 3rd generation. LHMs, the experimental males are trending towards being better. And then 5 or 6 generations later, we did this N study, and we saw that IV were pretty equal, but the LHM experimental males were actually a bit more fit than the controls. Then looking at female fitness, same thing here, except for its number of offspring produced. So you see that both control females started out doing better. Then we see experimentals actually doing better near the end. And then similar with the LHMs, experimental doing better. Interestingly, at the end, we saw a total switch in the IVs. Were controlled and much better, and LHMs kind of equaled out. This is relatively new data to us. We're sort of thinking there's something sexually antagonistic factors going on here, because you can see here where the LHM females are doing really well on the right side here. The males aren't doing great, and you can see when they do really well here, the females kind of end up doing not so great. So we think when males do well, females do poorly. When females do well, males do poorly. This is the behavioral assay. So this is the food preference. So this is IVs on this slide only, control on the top, experiment on the bottom, females pink, males blue. So we thought we'd see the same preference for protein for females, carbs for males like Delisle saw. And we did see that. We saw the separation, but we saw it in both control and experimental. We saw it more so in experimental, but we did see a specialization in both. And then similarly with the LHMs, we saw the same thing. We saw a separation over time, more so in experimental than control, which is interesting. But we sort of think we predicted increasing your choosiness would increase your fitness. But now we kind of think it's some sort of feedback loop where increasing fitness or increasing choosiness increases fitness, which increases choosiness, which increases fitness. And due to them having different environments to what they're usually in, it's kind of just a better environment overall. So even the controls did better, which led to this increase in choosiness. But we do see more of this increase in the experimental, which is interesting. Overall, we saw an increase in fitness for both males and females, but it was different for the LHMs and IVs, which is what we wanted. That's why we did the two different populations. We wanted to see the universality of this and how it affects different populations. So that was really interesting. Um, this shows it's kind of more complex than what Dalal saw, where he saw that competition really affected this evolution. Where if we take that out, it's a bit more, there's more factors than just competition that affect this. But it is some evidence for this diet partitioning influencing sexual dimorphism and fitness. But being a experimental evolution experiment, there's like a million ways we can go with this. In the future, we know mated flies have different diet preferences to virgin flies, so that's a thing to look at. We saw Delisle's low nutrition play a part in this, so what if we combined our methodologies, see how that plays out. I had an undergrad look into social interaction, and he found some interesting things because presence of conspecifics actually does influence your diet choice. Um, so that's interesting. Jen Perry's lab also looks at this too. There's some cool stuff there. And again, 13 generations, is that enough? Does this plateau? Does this increase going up, like going more? A lot of questions. But I think this is significant because it does provide some evidence for this ecological sexual dimorphism arising, which can be applied to conservation biology or animal husbandry or these captive breeding programs where the fitness of a population is really important, and you need to know if there are these female and male specific diet preferences that will benefit them. But yeah, I'd like to thank my committee and everyone in the Long Lab for all of their help. And I have any questions?
Thank you,
And thank you for
Owen.
listening. Thank you.
I have a question.
Yeah.
So, you're looking at flies here, but I'm curious, like in the last few slides, you actually put up like photos of birds. So I'm wondering, like, how feasible do you think this kind of the same logic of
Hmm.
experiments that you're doing here can be also applied to, say, like I think more complex organisms like birds?
Yeah, good point. Yeah, so when Darwin first mentioned this, he even said we see this more in lower life forms. Like he mentioned some like mosquitoes where females have these they can actually take blood meals or males feed on nectar and stuff. But we do see it in birds. We saw it in the oyster catchers. There's that now extinct. I think it's Huia bird. I think it's in New Zealand where they had really distinct male and female bill dimorphisms. Um, there's new studies that look at kind of why this is rare, and it's kind of there's these really specific parameters that be met. It basically depends on mating behavior, like if it's more monogamous species are more susceptible to this evolution. There needs to be like certain resources on either end of the spectrum that go underutilized so they can take advantage of it, and a lot of other things. So it's quite specific parameters for this to happen. But we do see it in birds and we see in other stuff like that. So it's definitely possible. Um, you know, um, but yeah.
All right. If no one has any more question, I think that will be the end of the second session of behavior.